Hi, this is the first webinar on a series of webinars on osteopathic themes by myself. You can subscribe to future webinars on my YouTube page, of which you see the URL here below. Let's start. First webinar on fat tissue and its influence on health and healing processes from an osteopathic perspective. In this session, we will explore how adipose tissue affects the body and how we can optimize treatment outcomes in patients with excessive fat tissue. The introduction. In clinical practice, we encounter patients with very different health profiles. Some are in relatively good condition, while others present with overweight and excessive fat tissue. We often observe that treatment outcomes are less effective in patients with increased body fat. Adipose tissue, normal or hyper, has its own metabolism, function and physiology, and has a strong influence on health and the self-healing mechanisms. This leads us to an important clinical question. What role does adipose tissue play in health, and how does it influence healing processes? Well, there are different types of fat tissue, different tissues. So the different tissues are the white adipose tissue, WAT, which consists out of subcutaneous adipose tissue and visceral adipose tissue. There is also brown adipose tissue, BAT, and a combination of the two, the beige adipose tissue. Adipose tissue is recognized as a complex and metabolically active organ that plays important roles in numerous physiological processes and responds rapidly and dynamically to changes in nutritional status. Not all fat is equal. Visceral fat located around the organs is more harmful than subcutaneous fat under the skin. Fat distribution is therefore more important than total fat mass. Fat tissue has its own metabolism and influences many bodily systems such as the respiratory system, the vascular system, the neurological system, the pain mechanisms, the immune system, and the self-healing system. Impaired adipose tissue function, rather than the absolute quantity of adipose tissue, may play a central role in the pathophysiology of metabolic disease risk. When adipose tissue becomes dysfunctional, it promotes chronic low-grade inflammation. This inflammatory state interferes with healing processes and with metabolic regulation. As fat tissue expands, oxygen supply becomes insufficient. This leads to tissue hypoxia, which further increases inflammation and metabolic dysfunction. Adipose tissue, the definition. Adipose tissue is a connective tissue found throughout the body. It is located under the skin, around organs, and within bone marrow and other tissues. Adipose tissue cellular composition. Adipose tissues are composed of multiple cell types. These include adipocytes, immune cells, vascular cells, and neural components. Together they form a complex and dynamic organ. The difference between subcutaneous and visceral fat. Subcutaneous fat is located beneath the skin. Visceral fat surrounds internal organs and is more strongly associated with disease, especially low-grade inflammation, chronic inflammation, and metabolic dysfunction. In addition to these major depots, adipose tissue is also found in several other anatomical locations, including the bone marrow, mammary glands, intermuscular regions, pericardial and epicardial areas surrounding the heart, the orbital cavities, as well as in specialized fat pads located in the palms of the hands and the soles of the feet. Another specialized form of adipose tissue is brown adipose tissue, BAT, which is primarily involved in thermogenesis and particularly abundant during infancy and then gradually decreases with age. In adults, it is mainly localized in regions such as the supraclavicular area, the upper back, and along the paravertebral regions of the spine. Here you see an image of baby fat, brown fat, and the remains of it in an adult, for example, as here in this picture, the upper part of the thoracic spine. Mobilizing and addressing adipose tissue can improve outcomes in patients with pain and chronic inflammation. Adipocytes. Here you see an image, a picture of an adipocyte. You see the nucleus. You see the fat reservoir, which can be more expanded or less expanded. You see the membrane, the cytoplasm, the Golgi apparatus, and of course the mitochondria, which provide the energy for the fat cell. Adipose tissue is crucial for health. In healthy individuals, total body fat typically ranges between approximately 10 and 35% of body mass, with variations, of course, depending on age, gender, and physiological status. In obesity, fat cells enlarge to store excess energy. This process is known as hypertrophy. Enlarged fat cells promote inflammation, insulin resistance, and metabolic disease. As we already said, adipose tissue is crucial for health, even in very thin patients. Too little fat tissue can also be problematic. Fat is then stored in organs, leading to metabolic dysfunctions. Lipodystrophia. Hypertrophic adipocytes are associated with chronic low-grade inflammation, altered adipokine secretion, and impaired insulin signaling, which contribute to the development of metabolic disease such as type 2 diabetes, cardiovascular disease, and non-alcoholic fatty liver disease. Conversely, insufficient adipose tissue can produce similar metabolic disturbances. In conditions characterized by reduced adipose mass, such as lipodystrophia, the body lacks adequate storage capacity for circulating lipids. As a result, lipids accumulate ectopically in non-adipose tissues, including the liver, skeletal muscles, and pancreas, leading to lipotoxicity and metabolic dysfunction. Some examples of lipodystrophia. You see that the fat tissue is then localized in body structures that normally do not contain that much lipid or that much fat. Adipocytes, fat cells, in humans have an average lifespan of approximately 10 years. During weight loss, adipocytes decrease in size. However, the total number of adipocytes remains stable. As a result, these cells retain the capacity to store lipids again if energy intake exceeds expenditure. Adipose dysfunction. Dysfunctional fat tissue is characterized by inflammation, hormonal imbalance, and metabolic disruption such as insulin resistance. Let's have a look at the body shape. Body fat distribution influences the risk for other diseases. Apple-shaped bodies have a higher risk than pear-shaped bodies. Pear-shaped pattern of fat distribution is often associated with a more favorable metabolic profile. Here in these images you see the difference. So apple body shape is more abdominal fat, and then the pear shape is more the fat around the pelvic area. Genetic factors play a significant role in determining an individual's baseline body type and overall body shape. However, lifestyle factors including dietary habits, physical activity and other health-related behaviors remain important determinants of overall health and body composition within the limits set by genetic background. Some recommendations: maintain a balanced diet, reduce saturated fats and limit refined carbohydrates. Regular physical activity is also essential. Let's now talk about the basic physiology of adipocytes. So there are different types of adipocytes. White adipocytes store energy. Brown adipocytes generate heat, and beige adipocytes can switch between the two functions. In newborn, around 5% of the body weight is made up of brown fat. It tends to be located on the upper half of the spine and towards the shoulders. It is active in the first 6 months of life and gradually decreases during the first year. A small amount persists into adulthood. You also see on the picture on your right side that the brown fat cell has more mitochondria, which means that these type of cells use more energy. Now, something about the adipose tissue cellular heterogeneity. Subcutaneous fat under the skin and visceral fat around the organs differ significantly in insulin sensitivity, metabolic activity and association with disease. Visceral fat is more linked to cardiometabolic disease. Different adipocyte subtypes specialize in lipid storage, signaling or in inflammation. Also, cell-to-cell communication between adipocytes, immune cells and endothelial cells is extensive. Adipose tissue contains different tissues. There are adipose progenitor cells. There are immune cells such as macrophages, M2, anti-inflammatory cells, or M1, which are pro-inflammatory cells, and they increase in obesity, for example, and there are also T cells and other immune cells. There are also vascular cells. They provide nutrients, they deliver oxygen, they form vessels. There are also fibroblasts in the matrix, which can lead to fibrosis, for example. And we see also neural components, sympathetic or fibers for thermogenesis. Adipose tissue should be considered as a multicellular metabolic organ, where adipocytes interact with immune, vascular, neural, and stromal cells to regulate energy homeostasis, inflammation, thermogenesis, and also endocrine signaling. Now let's talk about lipogenesis and lipolysis. Lipogenesis is the metabolic process of synthesizing fatty acids and triglycerides, fats, from non-fat sources like carbohydrates, primarily occurring in the liver and adipose tissue. It is stimulated by high carbohydrate intake. It is suppressed by polyunsaturated fatty acids and also by fasting. Lipolysis is the breakdown of fat into usable energy. It is activated during fasting and during exercise. Fat accumulation in adipose tissue is determined by the balance between lipid synthesis, which is lipogenesis, and lipid breakdown, including lipolysis and fatty acid oxygenation. This process is strongly influenced by dietary composition. Let's look at the hormonal regulation. Hormones regulate fat metabolism. Certain hormones inhibit lipogenic activity, such as leptin, whereas others, including angiotensin, can promote lipid synthesis. Additionally, glucose acts as a direct substrate for lipogenesis and indirectly enhances this process by stimulating insulin secretion and suppressing glucagon release from the pancreas. Now, fat cells produce several hormones and signaling molecules. Fat cells produce hormones known as adipokines. These regulate appetite, metabolism, and even inflammation. Here you see on this image, you see several hormones produced by fat cells. Again, here you see that the fat cells produce several hormones and signaling molecules. They regulate appetite, they have an influence on the immune system, on the reproduction system, on the glucose and lipid metabolism, on angiogenesis, on fibrinolysis, coagulation, body weight homeostasis, and also on the vascular tone control. White adipose tissue, WAT, is considered one of the largest endocrine organs in humans. Its functions arise from the capacity of adipocytes to secrete a wide range of bioactive molecules, including hormones, growth factors, enzymes, cytokines, complement components, and extracellular matrix proteins. These adipokines, particularly in the regulation of numerous physiological processes. In addition, adipose tissue expresses receptors for many of these signaling molecules, enabling autocrine, paracrine, and endocrine communication. Through these mechanisms, white adipose tissue contributes to the regulation of food intake, energy expenditure, metabolic homeostasis, immune responses, and blood pressure regulation. Adipose tissue is dynamically involved in cell function regulation through a complex network. And this network consists of endocrine, which signals travel through the circulatory system to reach all parts of the body, paracrine, which signals are sent only to cells in the vicinity of the cell station, and autocrine, only affecting cells that are of the same type signals. That influences the response of many tissues, including hypothalamus, pancreas, liver, skeletal muscle, kidneys, endothelium, and the immune system, amongst others. Leptin is one of the hormones produced by the fat cells. Leptin is a peptide hormone primarily secreted by adipocytes and encoded by the obese gene. Leptin is a crucial hormone that informs the brain about energy stores and regulates satiety. Although leptin is classically known for its role in regulating appetite, neuroendocrine function, and energy homeostasis through signaling pathways in the hypothalamus, it is now recognized to influence a wide range of additional physiological processes. These include the regulation of metabolic pathways, endocrine signaling, and immune function. Furthermore, emerging evidence suggests that leptin may participate in other biological processes that remain to be fully characterized. This regulation of leptin signaling has been associated with several metabolic disorders, particularly obesity and components of the metabolic syndrome. Circulating leptin concentrations are typically higher in females than in males, which can be partly explained by hormonal regulation. Androgens inhibit leptin production, whereas estrogens stimulate its expression, and adipose-specific differences in leptin synthesis also contribute to this variation. Leptin production is greater in subcutaneous adipose tissue than in visceral adipose tissues. Now there is also leptin resistance. Now, leptin dysfunction in obesity, primarily leptin resistance, occurs when the brain becomes less responsive to the satiety hormone produced by adipocytes. This impaired signaling can lead to persistent hunger, a reduced metabolic rate, and subsequent weight gain. The symptoms of leptin resistance: increased appetite, food cravings, and hunger even after eating. Cravings for sugary or high-fat foods, fatigue or low energy levels. Weight gain despite adherence of dietary control and regular physical activity. Difficulty in losing weight or an inability to reduce body weight despite a calorie-restricted diet and consistent exercise. Persistent hunger and food cravings even after eating. Cravings for energy-dense foods, particularly those high in sugar or fat. Fatigue or reduced energy levels. Sleep disturbances, including difficulty falling asleep or insomnia. Mood fluctuations or increased irritability. Cognitive difficulties such as impaired concentration, which is sometimes called brain fog. Joint pain or systemic inflammation, and elevated blood pressure or increased cholesterol levels. So leptin resistance is something that we see very often in our practices. The treatment for leptin resistance has to do with the thyroid gland. So there is a need of thyroid treatment. There is also the need for dietary modifications, and intermittent fasting here plays an important role. Physical activity, of course, is also important, just as adequate sleep, and we also can use supplements such as vitamin D and or probiotics. Stress reduction is, of course, as always, very important. It is critical to maintain leptin balance, as low levels can lead to starvation and heightened infection risk, while high levels can trigger insulin resistance, metabolic syndrome, obesity, and inflammation, thereby escalating cardiovascular risk. Leptin levels are directly proportional to the percentage of body fat mass. Consequently, circulating leptin concentrations are generally higher in women than in men, which can be attributed to women's higher body fat percentage and differences in hormonal profiles, particularly higher estrogen levels in women compared with androgens in men. Here on these pictures you see also a difference in distribution of subcutaneous fat in males and in females. Then there is another hormone which is produced, ghrelin. Ghrelin is an important hormone. It is often referred as the hunger hormone. It is a peptide hormone primarily produced not necessarily by the adipocytes but by the stomach, especially during fasting or when the stomach is empty. It stimulates appetite, increases food intake, and promotes fat storage by transmitting signals to the brain, particularly to the hypothalamus. Here you see an image of the hypothalamus. Still about ghrelin. In addition to its role in appetite regulation, ghrelin also contributes to the stimulation of growth hormone secretion and plays an important role in the regulation of energy balance. Higher circulating levels of ghrelin are associated with increased sensation of hunger, whereas lower levels are linked to greater satiety and reduced caloric intake. Consequently, strategies that help reduce ghrelin levels may facilitate appetite control and support weight management. Ghrelin levels are at their highest just before a meal and then subside for about 3 hours after a meal. Foods that suppress appetite generally fall into 3 main categories: high volume, low calorie foods, high protein foods, and foods that are rich in healthy fats. High volume, low calorie foods promote satiety by increasing gastric distension, which stimulates mechanoreceptors in the gastrointestinal tract and triggers satiety signals to the brain. High protein foods can suppress appetite through hormonal mechanisms, including reduction in circulating ghrelin levels and increases in satiety-related hormones. Not all high-fat foods exert the same satiety effects. Those rich in unsaturated fatty acids are generally considered more beneficial in this context. Additionally, certain foods contain bioactive compounds with potential appetite suppressing properties, Examples include cayenne pepper and ginger, which have been associated with modest effects on appetite regulation. Ghrelin is involved in a wide range of physiological processes besides the regulation of appetite and energy balance. It also promotes adipose tissue storage, it inhibits insulin secretion, and it stimulates growth hormone release. In addition, ghrelin has been implicated in reward-related processes, increased gastric acid secretion, enhanced gastrointestinal motility, and several other potential physiological effects. Now there is also ghrelin resistance. Ghrelin resistance is a metabolic condition commonly observed in obesity, in which the body exhibits a reduced physiological response to ghrelin, the hormone responsible for stimulating appetite. Despite potentially lower circulating ghrelin concentrations in individuals with obesity, the central nervous system may become less responsive to its regulatory signals. This impaired signaling can contribute to dysregulated appetite control, altered energy storage, and difficulties in achieving or maintaining weight loss. The treatment for ghrelin resistance. Consuming balanced meals with an adequate protein content. Maintaining a consistent and regular eating schedule or pattern. Ensuring sufficient sleep, as sleep deprivation is associated with increased ghrelin levels. Managing physiological stress, since chronic stress can influence appetite-regulating hormones. Engaging in regular physical activity, particularly moderate intensity aerobic exercise and resistance training. Something about the relation between leptin and ghrelin hormones. Leptin and ghrelin are key hormones in regulating energy balance. Leptin functions primarily as a mediator of long-term energy homeostasis by suppressing appetite and reducing food intake, thereby contributing to the regulation of body weight. In contrast, ghrelin acts as a short-term, rapidly responding hormone that stimulates appetite and is involved in the initiation of meals. In individuals with obesity, circulating levels of the anorexigenic hormone leptin are typically elevated, whereas levels of orexigenic hormone ghrelin are often reduced. Despite the increased leptin concentrations, obese individuals commonly exhibit leptin resistance, a condition in which central leptin signaling is impaired and appetite suppression is diminished. Another hormone is adiponectin. Adiponectin is an important hormone produced by adipose tissue that plays a key role in regulating glucose metabolism, fatty acid oxidation, insulin sensitivity, and anti-inflammatory processes. It enhances metabolic efficiency and is considered crucial for maintaining cardiovascular health. Higher circulating levels of adiponectin are typically associated with a healthy body weight and favorable metabolic status, whereas lower levels are commonly linked to obesity, type 2 diabetes, and cardiovascular disease. A bit more about adiponectin. The function is adiponectin exerts anti-inflammatory effects, enhances insulin sensitivity, and promotes fatty acid oxidation. The production, it is primarily secreted by white adipose tissue. And there is a relationship with adiposity in contrast to many other adipokines. Circulating adiponectin levels decrease as body fat mass increases. There are also health benefits. Adiponectin has protective effects against cardiovascular disease, particularly arteriosclerosis, and it contributes to the maintenance of metabolic homeostasis. The regulation, regular physical activity, and a healthy dietary pattern have been shown to increase circulating adiponectin levels. Adiponectin exerts anti-inflammatory effects in several cell types, including macrophages, a type of white blood cells involved in the recognition and elimination of pathogens such as bacteria and viruses, the endothelial cells, which form the inner lining of the heart and blood vessels and play a key role in vascular function, then also the skeletal muscle cells, where adiponectin contributes to the improved metabolic and inflammatory regulation. Then also the epithelial cells, which form protective layers covering the internal and external surfaces of the body. Lifestyle interventions such as regular physical activity and gradual healthy weight loss have been shown to increase circulating adiponectin levels. However, individuals should consult a healthcare professional before making significant changes to their diet or exercise regimes to ensure that these modifications are appropriate for their health status. Let's see how fat cells work. Now, during periods of fasting, when no food is being absorbed from the gastrointestinal tract, circulating insulin levels decrease due to the absence of nutrient-induced insulin secretion. Nevertheless, the body continues to require energy to sustain essential physiological processes. Under these conditions, energy must be mobilized from internal reserves, including stored glycogen, lipids, and proteins. To maintain energy homeostasis during fasting, several endocrine organs release hormones that promote the mobilization and utilization of stored energy substrates. The pancreas. The pancreas secretes glucagon, which simulates glycogenolysis and gluconeogenesis in the liver. The anterior pituitary gland releases growth hormone, which promotes lipolysis and reduces glucose uptake in peripheral tissues. Also the anterior pituitary gland, because this gland secretes adrenocorticotropic hormones, which stimulates the adrenal cortex to produce glucocorticoids. Also the adrenal medulla, because this organ releases adrenaline, which enhances glycogen breakdown and stimulates lipolysis. Also the thyroid gland, because this organ produces thyroid hormones which increase basal metabolic rate and regulate overall energy metabolism. Together, these hormonal responses coordinate the mobilization of stored nutrients and ensure a continuous supply of metabolic fuel to vital organs during fasting. These hormones act on hepatocytes, skeletal muscle cells and adipocytes, producing metabolic effects that largely oppose those of insulin. Now, during fasting, during periods of fasting or during physical exercise, when external nutrient supply is limited, the body must rely on endogenous energy stores to maintain adequate energy production. Glucose represents the primary metabolic fuel for many tissues. Certain cell types, particularly neurons in the brain, depend almost exclusively on glucose as an energy source under normal physiological conditions. Consequently, maintaining blood glucose levels is a critical component of metabolic homeostasis. The first mechanism for maintaining energy availability involves the breakdown of stored carbohydrates in the form of glycogen. This process, known as glycogenolysis, converts glycogen into glucose molecules that can be released into the bloodstream. When glycogen stores become depleted, the body increasingly relies on lipid metabolism. Stored triglycerides in adipose tissues are hydrolyzed through the process of lipolysis, producing glycerol and free fatty acids. Free fatty acids can be transported to peripheral tissues where they undergo beta oxidation to generate ATP, energy. Alternatively, substrates derived from lipid metabolism can contribute to gluconeogenesis, a metabolic pathway in which glucose is synthesized from non-carbohydrate precursors. Within adipocytes, several lipolytic enzymes catalyze the hydrolysis of triglycerides into fatty acids and glycerol. These enzymes are activated by hormones such as glucagon, epinephrine, and growth hormones. The resulting glycerol and free fatty acids are released into the circulation and they are transported to the liver. Once in the liver, glycerol can serve as a substrate for gluconeogenesis, while fatty acids may undergo oxidation for energy production or contribute indirectly to glucose synthesis through intermediary metabolic pathways. Let's talk about losing weight and losing fat. Your weight is determined by the rate at which you store energy from the food that you eat and the rate at which you use that energy. Remember that as our body breaks down fat, the number of fat cells remains the same. Each fat cell simply gets smaller. Fat cell memory and the yo-yo effect. Reducing body weight to improve metabolic health and obesity-related comorbidities is a central objective in the treatment of obesity. However, long-term maintenance of weight loss remains a major challenge, as the body appears to retain an obesogenic memory that resists sustained reduction in body weight. The epigenetic memory appears to predispose adipocytes to future transcriptional dysregulation when exposed again to a high-fat diet. This suggests that stable epigenetic changes in adipocytes and potentially in other cell types may underlie the persistence of obesogenic memory. Collectively, such cellular programming may prime adipose tissue for maladaptive responses in obesogenic environments, thereby contributing to the well-known yo-yo effect frequently observed during repeated cycles of dieting and weight regain. Human fat cells live for around 10 years, which means that it could take 10 years for the obesity memory in cells to vanish. Fat cells cannot be destroyed through natural means like diet and exercise. It's not possible. While healthy habits shrink fat cells by emptying their stored triglycerides, the cells remain in the body and they can easily expand again. Natural, permanent removal is not possible. Instead, cells shrink with weight loss. Let's talk about fat tissue and aging. Aging is a biological process characterized by the progressive loss of physiological integrity and the gradual decline of organ function, ultimately increasing the risk of mortality. Aging is frequently associated with obesity and metabolic dysfunction, including the development of insulin resistance. Recent studies suggest that age-related insulin resistance is linked to immunosensogenic and inflammaging. Now, the term inflammaging describes the chronic, low-grade inflammation state that accompanies the aging process. This condition reflects a complex imbalance between pro-inflammatory and anti-inflammatory responses and is largely mediated by immune cells, particularly macrophages. Inflammaging affects multiple tissues and organs, including adipose tissue, and contributes to metabolic dysregulation associated with aging. With advanced age, adipose tissue undergoes several structural and functional alterations. Changes in fat distribution, more in the abdominal region, ectopic lipid depositions, dysregulation adipokine secretion, vascular changes, structural changes, and reduced oxygenation. Macrophages in young, obese, and aged adipose tissue. In young adipose tissue, most adipose tissue macrophages exhibit an M2 phenotype. These macrophages perform several functions beyond their anti-inflammatory role, including efferocytosis, lipid buffering, angiogenesis, and regulation of iron homeostasis. Through these activities, M2 macrophages contribute to the maintenance of adipose tissue homeostasis. Aging is a chronic and complex physiological process that progressively disrupts energy homeostasis. In aged adipose tissue, the dynamic balance between lipid storage and lipolysis becomes poorly regulated. In aged adipose tissue, the total number of ATMs does not necessarily change substantially. However, the ratio of M1 to M2 macrophages increases. Aging therefore shifts the balance of adipose tissue macrophages towards a pro-inflammatory M1 dominant phenotype. As a result, the mobilization of free fatty acids is dysregulated, contributing to increased visceral adiposity, reduced physical performance, and decreased tolerance to cold. Adipose tissue macrophages have been implicated in the age-associated decline in lipolytic activity. In obesity, the saturated fatty acid palmitate can promote macrophage retention in adipose tissue by increasing the expression of netrin-1. This mechanism contributes to the persistence of macrophage accumulation within obese adipose tissue and is associated with the development of insulin resistance. There is also an adipose-brain connection, or sometimes also called the adipose-brain axis. Now, the central nervous system and adipose tissue communicate through complex bidirectional signaling pathways that regulate multiple metabolic functions. The hypothalamus, a key homeostatic region of the brain, integrates both exteroceptive and interoceptive signals to regulate appetite, energy expenditure, and glucose and lipid metabolism. Part of this regulation occurs through neural control of white adipose tissue and brown adipose tissue. Now, in this adipose-brain connection, the autonomic nervous system plays an important role. Neural innervation, particularly through the sympathetic and parasympathetic nervous system, plays an important role in modulating adipose tissue activity. White adipose tissue is directly innervated by sympathetic nerve endings of the autonomic nervous system. Upon binding norepinephrine to beta-adrenergic receptors on adipocytes, a signaling cascade is initiated that stimulates lipolysis and promotes the release of free fatty acids and glycerol from white adipocytes. White adipose tissue consists of multiple fat depots, including subcutaneous and visceral adipose tissue. Each of these depots is characterized by distinct patterns of sympathetic innervation density, differential receptor expression, and varying levels of sympathetic nerve activity, reflecting functional heterogeneity amongst adipose tissue depots. A crucial function of sympathetic innervation of white adipose tissue is controlling lipolysis. Importantly, sympathetic innervation also plays a critical role in the development and maintenance of white adipose tissue by regulating the entire life cycle of pre-adipocytes, including their proliferation, differentiation, and apoptosis. The paraventricular nucleus of the hypothalamus receives inputs from several hypothalamic nuclei, including the suprachiasmatic nucleus, which functions as the brain's primary circadian clock, and the arcuate nucleus. The PVH sends autonomic projections to the dorsal motor nucleus of the vagus nerve, where parasympathetic motor neurons that innervate the white adipose tissues are located. In addition, the PVH projects to the intermediolateral column of the spinal cord, which serves as the source of sympathetic innervation of white adipose tissue. I repeat this sentence for osteopaths because we very often use mobilization and manipulation of specific segments. So the PVH projects to the intermediolateral column of the spinal cord, which serve as the source of sympathetic innervation to the white adipose tissue. In contrast to the well-established sympathetic innervation of white adipose tissue, which has been confirmed by diverse research groups and study models, the functional outcome of parasympathetic nervous system innervation of white adipose tissue remains controversial, with different studies reporting conflicting results. Parasympathetic innervation of white adipose tissue does not appear to play a major role in lipogenesis, which might have been expected as a physiological counterpart to the lympholytic activity mediated by the sympathetic nervous system. Instead, it is demonstrated that parasympathetic innervation of white adipose tissue is involved in the regulation of leptin and resistin production, as well as insulin sensitivity. This was evidenced by observations that vagotomy resulted in decreased insulin-dependent glucose uptake and reduced free fatty acid uptake in adipose tissue. These neural pathways influence key processes in white adipose tissue and in brown adipose tissue, including lipolysis and thermogenesis, thereby contributing to overall energy balance. Adipose tissue itself also functions as both an energy storage organ and an endocrine organ, secreting hormones and signaling molecules that can influence brain function and systemic metabolic regulation. Recent research has identified additional communication mechanisms between the brain and adipose tissue, including extracellular vesicles and neuromesenchymal units, which add further complexity to this regulatory network. Disruption in brain adipose tissue communication have been implicated in metabolic disorders such as obesity and type 2 diabetes. Understanding these signaling pathways may therefore provide new opportunities for the development of therapeutic strategies aimed at improving metabolic health. One of the principal regulatory pathways of adipose tissue is the sympathetic nervous system, which transmits signals from the brain to adipocytes and other adipose tissue cells through the release of catecholamines. These signals play a central role in the regulation of adipose tissue metabolism. Sympathetic nervous system mediated signaling influences several key functions of adipose tissue, including thermogenesis, neutral lipolysis, and the activity of immune cells within the tissue. Sympathetic output, primarily through catecholamines such as norepinephrine, can limit adiposity by directly stimulating adipocytes, thereby promoting the mobilization and utilization of stored lipids. Interestingly, it has been reported that the brain regulates leptin gene expression in white adipose tissue through sympathetic innervation. Lesions of the suprachiasmatic nucleus, which disrupts the central circadian clock, abolish the normal daily rhythm of leptin secretion. In addition, sympathetic nervous system innervation has been shown to regulate the life cycle of pre-adipocytes, including their proliferation. In response to external stimuli such as cold exposure, adrenaline-mediated activation of the sympathetic nervous system promotes the browning of white adipocytes. During this process, white adipose cells acquire characteristics like those of brown adipose tissue, enabling them to dissipate energy as heat through thermogenesis. Recent studies also suggest that thyroid hormones play an important role in promoting the browning of white adipose tissue. However, this mechanism does not necessarily depend on sympathetic nerve system activation. Collectively, these findings highlight the crucial role of neural innervation in maintaining adipose tissue metabolic health across multiple levels, including functional regulation, tissue development, and cellular lineage specification. Brown adipose tissue is the primary site of non-shivering thermogenesis, with the capacity to increase energy expenditure by approximately 5 to 10%, oxidize intracellular triglycerides, and enhance glucose uptake upon activation. Brown adipocytes are characterized by an abundance of mitochondria and multilocular lipid droplets, enabling them to play a central role in adaptive thermogenesis through the expression of uncoupling protein 1. The interaction between the central nervous system and brown adipose tissue facilitates an effective physiological response to metabolic changes such as fasting, cold exposure, and disturbances in energy balance. Communication between the brain and brown adipose tissue occurs primarily through the sympathetic nervous system. The pathway represents the principal efferent innervation of brown adipose tissue and, like white adipose tissue, is predominantly composed of tyrosine hydroxylase (TH) expression neurons. Neural activation of brown adipose tissue occurs in response to various stimuli, including cold exposure, thyroid hormones, and disturbances in energy balance, all of which stimulate the sympathetic nervous system. Signals related to cold exposure and energy imbalance are sensed and integrated by the brain, triggering the release of norepinephrine from TH-positive sympathetic neurons, which subsequently activates beta-3 adrenergic receptors. These receptors are highly expressed in brown adipose tissue and play a critical role in adaptive thermogenesis, with their sensitivity modulated by several factors, including thyroid hormones. The sympathetic nervous system activity involved in brown adipose tissue thermogenesis originates in the hypothalamus. Within this brain region, several hypothalamic nuclei integrate and coordinate signals that regulate body temperature and energy balance, highlighting the essential role of thermoregulation in maintaining metabolic homeostasis. Among the brain regions involved in the regulation of core body temperature, collectively referred to as the thermoregulatory pathway, the preoptic area is recognized as a key regulatory center. Here you see now, for example, an overview of the signaling, or let's say the relation between adipose and brain tissue. We see on the left side that the white and the brown adipose tissue send sensory information towards the brain, and then we see that there is efferent information that goes down, that comes down from the hypothalamic and extra hypothalamic areas, and that efferent information influences food intake, energy expenditure, glucose metabolism, insulin sensitivity, and even sympathetic innervation. And all of this influences body weight, lipid metabolism, glucose metabolism, sympathetic neurite outgrowth, and thermogenesis. Let's look at the endocrine regulation of the adipose brain axis. Adipocytes are regulated by multiple mechanisms, including neuronal, endocrine, and also metabolic signals. While insulin is the classical endocrine regulator of lipid metabolism, in adipose tissue, several other hormones also play a significant role in controlling adipose tissue physiology. The pituitary gland is often referred to as the master endocrine gland because of its central role in regulating numerous physiological processes. The anterior pituitary comprises a heterogeneous population of endocrine cells that are regulated by hypothalamic releasing and inhibiting factors delivered through the hypophyseal portal circulation. Let's then look at the feeding behavior in the adipose brain axis. Numerous molecules are involved in the regulation of feeding behavior, and both these molecules and their receptors are expressed in the hypothalamus of the brain as well as in adipocytes. Based on evidence suggesting a functional link between the central nervous system and adipose tissue, the concept of a brain adipose axis has been proposed. Regulatory effects on appetite within the brain, particularly in hypothalamic circuits, control energy balance. Secretion into the systemic circulation, allowing these molecules to act as circulating signaling proteins. These signaling molecules can generally be categorized into orexigenic, which is appetite stimulating, and anorexigenic, which is appetite suppressing groups. Within adipose tissue, most orexigenic molecules exhibit adipogenic activity, promoting fat cell development and lipid storage, whereas anorexigenic molecules typically inhibit adipocyte proliferation and fat accumulation. Although the precise mechanism through which these circulating factors coordinate communication between the brain and peripheral adipocytes are not yet fully understood, current evidence suggests the existence of a bidirectional feedback system between the hypothalamic signaling pathways and adipose tissue. Disruption or dysregulation of this brain adipose axis may contribute to the development of obesity and metabolic disorders such as the metabolic syndrome, as illustrated here on this image. There are even other connections between adipose tissue and the brain. For example, the hormone adiponectin. As mentioned before, another important adipocyte-derived hormone is adiponectin. Adiponectin exerts its effects through its receptors, AdipoR1 and AdipoR2, which are expressed in several nuclei of the mediobasal hypothalamus. Through these receptors, adiponectin influences a variety of physiological processes, including food intake, energy metabolism and body weight regulation, bone metabolism even, and glucose homeostasis. Adiponectin exerts either stimulatory or inhibitory effects on food intake by acting on pro-opiomelanocortin neurons, with these effects depending on the energy status of glucose concentration in the brain. Adiponectin also acts on oxytocin neurons, where it inhibits neuronal excitability, suggesting a potential role in regulating energy homeostasis through the modulation of pituitary hormone secretion. Furthermore, adiponectin influences hypothalamic circadian rhythms by upregulating the core clock gene BMAL1 via AdipoR1 signaling, thereby regulating the expression of orexigenic neuropeptides and food intake. Similar to leptin, adiponectin can also induce catabolic processes in astrocytes, thereby improving nutrient availability in the hypothalamus. Collectively, these findings indicate that adiponectin plays a critical role in regulating hypothalamic activity and systemic energy homeostasis through multiple mechanisms that influence both neuronal and non-neuronal cell populations. Another hormone also can play a role, resistin. White adipose tissue also communicates with the brain through resistin. And resistin is an adipokine produced by adipocytes in humans with circulating levels positively correlated with adiposity. The name resistin derives from its ability to promote insulin resistance. In addition to its role in insulin resistance, resistin influences metabolic regulation and inflammatory responses through activations in the hypothalamus. Within the central nervous system, resistin has been shown to increase sympathetic nerve activity, thereby influencing cardiovascular function, while simultaneously inhibiting sympathetic nerve activity directed towards brown adipose tissue. Evidence suggests a functional crosstalk between leptin and resistin in the regulation of metabolic processes, particularly through their actions in the hypothalamus. Studies indicate that resistin can attenuate leptin's effects on food intake and exert opposing effects on insulin sensitivity. Metabolic regulation of the adipose brain axis. We talk about fatty acids. White adipose tissue also communicates with the brain through the release of various lipid species into the circulation. Fatty acids. So fatty acids are released from adipocytes into the bloodstream, where they are commonly bound to albumin or incorporated as triglycerides within lipoproteins. During circulation, these fatty acids can cross the blood-brain barrier through multiple transport mechanisms, including passive diffusion and facilitated transport. The blood-brain barrier expresses several specialized transport proteins that facilitate fatty acid uptake into the brain. Once in the brain, fatty acids can be esterified or incorporated into membrane phospholipids. In addition, they can function as signaling molecules, either by binding to membrane receptors or by acting as intracellular messengers that modulate signal transduction pathways. One of the most studied metabolic roles of fatty acid sensing is the regulation of energy and glucose homeostasis. Specialized neurons in the hypothalamus detect fluctuations in fatty acid concentrations and adjust their activity accordingly, thereby coordinating behavioral and metabolic responses. Hypothalamic fatty acid sensing also influences the activity of several hormones. Fatty acid signaling within the hypothalamus regulates insulin secretion and action, a process that depends on fatty acids beta oxidation and the activity of the enzyme carnitine palmitoyltransferase 1, which controls the transport of fatty acids into the mitochondria. Also, the immune system plays a role in the regulation of the adipose brain axis. The innate immune system plays a critical role in the regulation of adipose tissue function. Amongst the various immune cell populations present in white adipose tissue, macrophages are the most abundant, accounting for more than half of the leukocytes in both lean and obese humans. Their accumulation within white adipose tissue is strongly associated with obesity. The existence of bidirectional communication between the central nervous system and resident immune cells in white adipose tissues contributes to the regulation of lipid homeostasis. Consequently, the immune system plays a crucial role in mediating the crosstalk between the brain and adipose tissue. In addition, cold exposure can stimulate macrophages to secrete signaling molecules that modulate sympathetic innervation of white adipose tissue. Now let's have a look at brown adipose tissue and the adipose brain axis. Traditionally, brown adipose tissue has been studied primarily for its central role in thermogenesis. However, in recent years, increasing attention has been directed towards the secretory function of brown adipose tissue and its role in interorgan communication. The molecules secreted by brown adipose tissue, often referred to as brown adipokines or bradykines, can act on distant organs and tissues, including the liver, the heart, skeletal muscles, white adipose tissue, and the brain. These signaling molecules exert their biological effects through autocrine, paracrine, and endocrine mechanisms. Adipose tissue-brain communication in health and in obesity. Let's compare this. The lean phenotype is characterized by optimal hypothalamic sensing, efficient thermogenesis in brown adipose tissue, and normal white adipose tissue function, collectively contributing to systemic metabolic homeostasis. The obese phenotype is associated with impaired hypothalamic sensing, which leads to reduced thermogenic activity in brown adipose tissue and dysfunctional white adipose tissue. This disruption results in decreased energy expenditure and altered adipokine secretion, including elevated leptin levels and reduced adiponectin concentrations. In addition, obesity is characterized by increased macrophage infiltration into white adipose tissue, promoting chronic low-grade inflammation and further metabolic dysregulation. At the neuronal level, the obese phenotype is marked by enhanced hypothalamic inflammation, which may lead to mitochondrial dysfunction, endoplasmatic reticulum stress, disrupted lipid metabolism, and impaired autophagy. Together, these cellular disturbances contribute to the development of leptin resistance. Also, the gut microbiota play a role, an important role, in the adipose tissue, gut adipose tissue axis. So the gut adipose tissue axis plays a critical role in the regulation of metabolic health. It represents a bidirectional communication network between the gastrointestinal tract and adipose tissue. This interaction influences several physiological processes essential for maintaining metabolic homeostasis, including energy balance, lipid metabolism, and inflammatory regulation. Communication between the central nervous system and the gut microbiota occurs through multiple pathways, including chemical, neural, immune, and endocrine signaling mechanisms. Alteration in the gut microbial composition, referred to as gut dysbiosis, can disrupt the bidirectional communication between the central nervous system and the gut microbiota and have been associated with the development of neurological and neurodevelopmental disorders. Consequently, the microbiota gut-brain axis has recently emerged as a potential therapeutic target for the treatment of metabolic and neurological disorders. However, although extensive experimental evidence from basic research supports the role of the MGBA, its translation into clinical application remains limited. Understanding the molecular interactions amongst the gut microbiota, the brain, and adipose tissue may provide novel therapeutic targets for the treatment of neurological disorders. Adipose tissue is composed of multiple cell types, including pre-adipocytes, mature adipocytes, fibroblasts, endothelial cells, and macrophages derived from the circulation. Now, the human digestive system contains approximately 10 trillion microorganisms, collectively carrying more than 9.9 million microbial genes. The gut microbiota performs numerous physiological functions, including the regulation of metabolism and nutrient absorption, maintenance of the integrity of the intestinal mucosal barrier, and modulation of immune responses. Obesity is closely associated with alterations in gut microbial composition, including the reduced microbial richness and diversity of a decrease in Bacteroidetes, one of the most abundant bacterial phyla in the gut. Changes in microbial composition are also linked to alteration in the gut microbial metagenome, particularly an enrichment of genes involved in energy harvest from the diet. The gut microbiota modulates host homeostasis through interactions with distal organs, including the liver and adipose tissue, primarily via the release of metabolites and cytokines. The liver communicates with the intestine through bile acids and also through various bioactive mediators. In addition, the gut microbiota contributes to the regulation of energy expenditure and body weight by promoting the browning of white adipose tissue, enhancing the activity of brown adipose tissue and influencing lipid metabolism. The gut microbiota also produces several microbial-derived metabolites and structural components which may play central roles in the development of inflammation, insulin resistance, and even obesity. Let's talk about intermittent fasting. Intermittent fasting is widely recognized as an effective strategy for weight loss and has been associated with multiple benefits for metabolic health. Recent studies suggest that intermittent fasting may induce the browning of white adipose tissue by altering the composition of the gut microbiota. An every other day fasting regime stimulates white adipose tissue browning and significantly improves obesity-related metabolic complications, including hepatic steatosis and insulin resistance. Therefore, understanding the interactions between the gut microbiota and adipose tissue, as well as the underlying mechanisms, may provide new strategies for the treatment of obesity and related metabolic disorders. People with obesity tend to have higher levels of the firmicutes and fewer bacteroidetes count than normal weight individuals. Let's talk about inflammation. Alteration in the abundance and diversity of the gut microbiota are closely associated with inflammatory processes in obesity. Individuals with approximately 40% reduced microbial gene richness exhibit more pronounced metabolic dysfunction and chronic low-grade inflammation. Conversely, an increase in microbial gene richness and diversity is associated with significant reductions in adipose tissue mass, circulating cholesterol levels, and inflammatory markers. Furthermore, obesity is characterized by shifts in the composition of specific microbial taxa. Probiotic strains can suppress adipose low-grade chronic inflammation by modulating the host's immune responses and inflammatory markers. Certain lactic acid bacteria strains have been shown to suppress chronic inflammation in adipose tissue by regulating the secretion of adipokines. Given their potential health benefits, probiotics have increasingly been incorporated into functional foods and therapeutic interventions. Lactobacillus particularly, species belonging to the Lactobacillus and Bifidobacterium genera, exhibit anti-inflammatory effects in adipose tissue. Let's now talk about adipose tissue oxygenation. First, in general. Adipose tissue oxygenation plays an important role in maintaining metabolic health. In obesity, rapid expansion of adipose tissue can outpace the development of adequate vascularization, leading to tissue hypoxia or reduced oxygen availability. Although adipose tissue is not a major consumer of oxygen compared with highly oxidative tissue such as, for example, skeletal muscle, adequate oxygen supply is still essential for maintaining normal cellular metabolism and adipocyte function. Insufficient vascularization and reduced blood flow in enlarged adipose depots can result in localized hypoxic conditions, particularly in hypertrophic adipocytes. Adipose tissue hypoxia has been shown to activate a variety of inflammatory and stress-related signaling pathways, including the stabilization of hypoxia-inducible factors, HIF. Cellular respiration. Cellular respiration is a series of metabolic reactions through which glucose and other energy substrates are oxidized to generate adenosine triphosphate, that's energy, the primary energy currency of the cell. The primary function of cellular respiration is to convert the chemical energy stored in nutrients, such as carbohydrates, proteins, and lipids, into a usable form of energy for the cell. This energy is generated in the form of adenosine triphosphate, ATP, which serves as the primary energy currency of the cell. ATP provides the energy required to drive numerous enderogenic biochemical reactions that would otherwise not proceed without an external energy input. These processes include muscle contraction, active transport across cellular membranes, protein synthesis, and cell division, amongst many other essential cellular functions. Cellular respiration consists of three main stages. There is the glycolysis, the citric acid cycle or the Krebs cycle, and there is the oxidative phosphorylation. White adipose tissue undergoes significant structural changes during obesity, most notably an enlargement of intracellular lipid droplets within the adipocytes. However, only the non-lipid cytoplasmatic components of adipocytes require oxygen for metabolic processes. Consequently, despite the increase in adipocyte size, the effective oxygen demand per unit of adipose tissue may not increase proportionally, as the lipid droplets itself is metabolically inert and does not consume oxygen. Adipose tissue oxygenation, however, so adipocytes receive oxygen primarily through the surrounding capillary network. However, in obesity, hypertrophied adipocytes can exceed the normal diffusion distance for oxygen, resulting in relative tissue hypoxia. This reduced oxygen availability can trigger inflammatory responses, promote macrophage infiltration into the adipose tissue, and contribute to decreased insulin sensitivity. Adipose tissue oxygenation varies between individuals, with lower oxygen levels commonly observed in people with obesity compared with lean individuals. Hypoxia in obesity. As adipocytes enlarge in individuals with obesity, they may outgrow their local vascular supply. This can lead to a reduction in the partial pressure of oxygen within adipose tissue, resulting in a state of relative hypoxia. Tissue pressure, oxygen pressure, reflects the balance between oxygen delivery and consumption. During the development of diet-induced obesity, adipocytes undergo significant structural and functional alterations, including cellular hypertrophy, which contributes to the expansion of adipose tissue and results in reduced efficiency of local blood flow. This condition may subsequently lead to tissue hypoxia, in which various cells within adipose tissue, such as adipocytes, immune cells, and endothelial cells, activate the transcription factor hypoxia-inducible factor, HIF, to adapt to the low oxygen environment. Consistent with this mechanism, visceral adipose tissue obtained from individuals with obesity has been shown to exhibit significantly reduced oxygen tension, accompanied by elevation expression of HIF1, alpha, and other genes associated with inflammation and fibrosis. The link between insulin resistance and cellular respiration. Reduced oxygen availability places adipocytes under metabolic stress and activates hypoxia-inducible factor 1 alpha. Activation of this pathway promotes inflammation signaling with adipose tissue and is associated with the development of insulin resistance. Effects of adequate versus reduced oxygenation. Under well-oxygenated conditions, adipocytes maintain normal metabolic function. In contrast, hypoxic conditions can promote adipocyte dysfunction and cell death, including necrosis and apoptosis, which may increase the release of free fatty acids into the circulation. Now, the increased metabolic demand in obesity. Although adipose tissue oxygenation is generally lower in obesity, the metabolic demand for oxygen at the cellular level may be elevated compared with lean individuals, further exacerbating the imbalance between oxygen supply and demand. The oxidation of fatty acids is an oxygen-dependent metabolic process. Adequate oxygen availability is required for mitochondrial oxidative phosphorylation, which enables the efficient breakdown of stored fatty acids and the production of ATP. The role of specialized adipose tissue. Brown adipose tissue differs functionally from white adipose tissue in that it contains a higher density of mitochondria and a more extensive vascular network. As a result, brown adipose tissue exhibits substantially greater oxygen consumption, which supports its primary role in thermogenesis and heat production. Oxygen fuels the mitochondria, the energy producers in the cell, allowing them to function at higher levels. This increased energy production leads to better fat metabolism, making it easier for your body to break down stored fat and use it as fuel. Oxygenation for enhanced fat burning. At the heart of hyperbaric oxygen therapy, we use this as an example, is delivering 100% pure oxygen to your body's cell in a pressurized chamber. The increased pressure allows your blood to carry more oxygen, which tissues throughout the body can use. This process isn't just great for healing, it's also beneficial for your metabolism. When exposed to extra oxygen, the body can burn fat more efficiently by boosting cellular activity. One of the major obstacles to weight loss is chronic inflammation, which can slow down metabolism and make it harder to shed pounds. Hyperbaric oxygen therapy promotes cellular healing and regeneration by improving oxygen supply to tissues, reducing oxidative stress, and aiding in the repair of damaged cells. This is particularly important for people struggling with weight loss, as inflammation is often tied to metabolic disorders like insulin resistance. By reducing inflammation, your body can better regulate hormones like insulin, which controls how your body stores and uses fat. Dense food components, such as fats and proteins, are not only low in oxygen content, but also require extra oxygen from the body to convert them into energy, which further depletes the body's oxygen reserves. Other oxygen-robbing foods include processed sugar, white flour, alcohol, and caffeinated drinks. Now, thus oxygen breaks down fat. Exercise speeds up the fat burning process because it pushes you to take more frequent, deeper breaths, and you need oxygen to burn fat. When your respiratory rate goes up, it also helps more carbon dioxide, one of the two byproducts of fat metabolism, to leave your body. Fat is converted to carbon dioxide and water. When you lose weight, your fat is actually exhaled as carbon dioxide. We breathe out fat. The rest is expelled as water through urine, sweat, and other body fluids. Almost everything we eat comes back out via the lungs. Every carbohydrate you digest and nearly all the fats are converted to carbon dioxide and water. The same goes, for example, for alcohol. Protein shares the same fate, except for the small part that turns into urea and other solids, which you excrete as urine. The only thing in food that makes it to your colon undigested and intact is dietary fiber. Everything else you swallow is absorbed into your bloodstream and organs, and after that it's not going anywhere until you've vaporized it. Fat metabolism and carbon dioxide production. So during the metabolism of fat, the carbon atoms stored in fatty acids are ultimately converted into carbon dioxide, which is expelled from the body through respiration. However, increasing breathing rate alone does not lead to weight loss. Hyperventilation or breathing excessively without an accompanying increase in metabolic activity does not increase fat oxidation and may lead to dizziness or fainting. The production of carbon dioxide during fat metabolism is directly linked to energy expenditure, which primarily increases throughout muscular activity. Physical movement elevates metabolic rate and thereby increases the rate at which substrates such as fats are oxidized. Even modest physical activity can significantly raise metabolic rate. For example, standing and performing routine activities such as dressing can more than double resting metabolic rate. Activities such as walking, cooking, vacuuming or sweeping can increase metabolic rate to approximately 3 times the resting level, thereby increasing oxygen consumption and carbon dioxide production. From a biochemical perspective, the oxidation of adipose tissue requires oxygen and produces carbon dioxide and water as metabolic byproducts. The complete oxidation of 100 grams of fat requires approximately 290 grams of oxygen and results in the production of about 280 grams of carbon dioxide and 110 grams of water. Consequently, the loss of body fat ultimately results in the release of carbon atoms as carbon dioxide during respiration. In practical terms, weight loss occurs when energy expenditure exceeds energy intake, regardless of the specific dietary composition. Any dietary pattern that provides less energy than the body expends will lead to a reduction in stored body fat. Obesity hypoventilation syndrome, OHS, obesity hypoventilation syndrome. Morbidly obese patients are in the group of patients who can desaturate fast because of changes in lung volumes and reduction in functional residual capacity due to obesity. Excess adipose tissue around the thoracic cavity increases the mechanical load on the chest wall and respiratory muscles, making it more difficult to achieve adequate lung expansion during inspiration or inhalation. This mechanical restriction reduces ventilatory efficiency and may impair the ability to maintain an appropriate breathing rate and depth. Consequently, ventilation becomes insufficient to adequately exchange gases, leading to elevated levels of carbon dioxide, which is then called hypercapnia, and reduced oxygen concentration, which then is called hypoxemia in the blood. The main symptoms of OHS are due to lack of sleep and include poor sleep quality, sleep apnea, daytime sleepiness, depression, headaches, and tiredness. Symptoms of low blood oxygen levels, called chronic hypoxia, and high carbon dioxide can occur. Symptoms include shortness of breath or feeling tired after little effort. OHS complications related to a lack of sleep may include depression, agitation, irritability, increased risk for accidents or mistakes at work, for example, problems with intimacy and sex, high blood pressure, right-sided heart failure, high blood pressure in the lungs. So, where do you now lose weight first on your body? The first place where you lose weight is often areas where fat is more metabolically active, like around your organs and upper body. After that, it may start to come off from longer-term storage areas, such as your hips, thighs, or lower belly. Several conditions affect brown fat in your body, including anorexia nervosa, congenital leptin deficiency, lipodystrophy, and lipoma. Obesity and the effect on the healing process. Obesity has a well-documented negative influence on the wound healing process, supported by a large body of biomechanical research. Excess adipose tissue alters immune function, blood supply, inflammation, and metabolic regulation, all of which are critical for normal tissue repair. Here are the main mechanisms identified in scientific studies. First of all, chronic low-grade inflammation. Obesity is characterized by chronic low-grade systemic inflammation. Enlarged adipocytes release pro-inflammatory cytokines which recruit macrophages into adipose tissue. These inflammatory mediators disrupt the normal phase of wound healing, delay the transition from inflammation to tissue repair, and impair fibroblasts. Studies show that obese individuals often have prolonged inflammatory phases, which slows tissue regeneration. Besides low-grade inflammation, there is also impaired blood supply and tissue hypoxia. As adipocytes enlarge in obesity, they can outgrow their blood supply, leading to reduced oxygen delivery. The consequences include tissue hypoxia, reduced angiogenesis, impaired collagen synthesis, increased risk of wound infection. Hypoxic adipose tissue activates hypoxia-inducible factor, which promotes inflammation and fibrosis, further impairing wound repair. And then there is also reduced immune function. Obesity alters immune cell activity and weakens host defense. Effects include dysfunctional macrophages and neutrophils, reduced pathogen clearance, increased susceptibility to infection, and because immune cells are essential in the early inflammatory stages of wound healing, impaired immune responses slow the healing process and increase complication rates. Then there is also the insulin resistance and the metabolic dysfunction. Obesity is strongly associated with insulin resistance, which negatively affects tissue repair. Insulin normally promotes cellular proliferation, protein synthesis, glucose uptake in healing tissue. When insulin signaling is impaired, fibroblasts' activity and collagen deposition decrease, slowing wound closure. And then there is also the mechanical stress on wounds. Excess body mass also produces greater mechanical stress on healing tissues, particularly after surgery. This can lead to wound dehiscence, that is reopening of wounds, increased tension on sutures, and delayed closure. And then there is also the altered adipokine signaling. Adipose tissue secretes hormone known as adipokines, such as leptin, adiponectin, resistin, and in obesity, adiponectin decreases. Leptin and pro-inflammatory cytokines increase. This hormonal imbalance contributes to increased inflammation, reduced tissue regeneration, and impaired vascular function. Summary. Obesity negatively affects wound healing through several interconnected mechanisms. Chronic inflammation, reduced tissue oxygenation, immune dysfunction, insulin resistance, mechanical stress on wounds, dysregulated adipokine signaling, And together these factors slow tissue repair and increase the risk of infection and post-operative complications. Here you see the cycle, for example, that start with bad nutrition, let's say. So high blood pressure, fatty liver disease, increases the risk of stroke, diabetes, can cause pregnancy problems, can cause kidney disease, sleep apnea will be there, osteoarthritis might follow, heart disease, an increased risk of developing cancer even. So all of these possibilities, negative possibilities, can be provoked by enlarged adipose tissue. Now, the osteopathic approach. How can we influence the fat metabolism? In general, we are osteopaths, and besides possible advice, we treat patients manually. This is extremely important in patients with enlarged white and brown adipose tissue, because without proper well-directed manual treatment to improve the quality, read oxygenation of the adipose tissue, treatment results will be poor or won't last for long. Very specific techniques must be used in well-determined treatment strategies. In different of my books, these specialized strategies and techniques are described with streaming videos. Now, how can we influence the fat metabolism? We have our osteopathic treatment, but of course there are also environmental factors. Environmental factors such as imbalanced diets, exposure to environmental toxins, and sedentary lifestyles can significantly impair organ function by inducing metabolic alterations, inflammation, and oxidative stress. These disturbances not only affect individual organs but also disrupt the communication pathways that coordinate systemic physiological processes, thereby triggering a cascade of adverse health outcomes. Excess energy intake promotes fat accumulation, primarily through adipocyte hypertrophy, which is associated with adipose tissue inflammation, fibrosis, and hypoxia, ultimately contributing to impaired metabolic health. Dysfunctional adipose tissue in this context also exhibits altered adipokine secretion, producing reduced levels of adiponectin, an insulin sensitizing adipokine, and elevated levels of leptin, which is secreted proportionally to fat mass. In obesity, increased circulating leptin levels are frequently associated with leptin resistance, a condition characterized by the diminished ability of the body to respond to the anorexigenic effect of this hormone. This condition arises through the chronic overactivation of leptin signaling in key hypothalamic regions, which subsequently trigger negative feedback mechanisms mediated by suppressor of cytokine signaling. Obesogenic diets also exert direct effects on the hypothalamus. Excessive consumption of dietary fat and sugar has been shown to induce mitochondrial dysfunction, endoplasmatic reticulum stress, impairment of autophagy, dysregulation of lipid metabolism, and inflammation within hypothalamic tissue. These alterations disrupt normal neuronal function and contribute to the development of obesity. Collectively, these mechanisms promote increased appetite and altered communication between the hypothalamus and adipose tissues, including white adipose tissue and brown adipose tissue. concerning the treatment, the osteopathic treatment. Therefore, I refer to the following two books. There is the book on nutrition and physical complaints, of which you can see the URL here, and the book on the metabolic syndrome. There are also other possibilities, and that is to follow one of my practical postgraduate courses where the manual techniques will be trained in detail. Besides the environmental factors, there is also the vascularization and oxygenation that we can treat as osteopaths. Oxygenation of the adipose tissues is important for its functioning. Mobilization of the adipose tissue is then a good option, but when there is not only hypoxia of the fat tissue, but also a general hypoxemia, hypoxemia must be addressed first. In the case of hypoxemia, there is a hierarchy strategy to be followed. Details on this strategy can be found in the book The Scientific Osteopathic Approach to Vascularization and Oxygen Supply in Patients. In that book you will see all the possible tests and techniques to differentiate hypoxia from hypoxemia, and all possible techniques to treat hypoxemia in a very efficient way. You can of course also follow one of my postgraduate courses to train all these techniques in detail. Important when we talk about vascularization and oxygenation is that treating hypoxemia is more important, has to be done before the treatment of local hypoxia. Then there are also the mechanical forces. Recently, research has been focusing on signaling mechanisms beyond nervous and chemical signals in the control of adipose brain axis. A hot topic in this area is the importance of mechanical forces, which are currently considered a key factor for the maintenance of white adipose tissue health. It is tempting to speculate that mechanical forces might control the production and release of adipokines, thereby playing a role in tissue communication. Adipocytes that contain too much fat particles will push against each other. This compresses the vascularization and neurology in between the fat cells. This compression phenomenon causes local ischemia, followed by local inflammation and followed by adhesions and scar tissue. Therefore, especially in the abdominal and pelvic region, we mobilize the visceral and fat tissues in all possible directions. We hereby avoid too much pain, not to provoke more inflammation. All tests and techniques on these fat tissues can be found in the book Scientific Approach to Patients with Abdominal Complaints. You can also, of course, as I told you before, follow one of my postgraduate courses to see all those tests and techniques in detail. Another element to influence fat metabolism is improving the quality of the cerebrospinal liquid or fluid. The aim by improving the quality of the cerebrospinal fluid is to improve the oxygenation of the brain tissue, especially in relation with central structures that influence fat tissue communication, such as the hypothalamus. Stasis of the cerebrospinal fluid. Stasis of that fluid is common, and it is detrimental to health. Mechanical forces. There are different influences that can cause cerebrospinal fluid stasis. cardiovascular influences. When the right atrium is weak or when there is heart failure, there is a reflux in the internal jugular vein, and this reduces the drainage of waste products from the brain. There are also respiratory influences, vasomotor influences, and mechanical influences. Degenerative changes or pathology of the brain is accompanied with stasis of the flow of the cerebrospinal fluid. Stasis seems to be particularly prone in the spinal canal. The presence gradient between the subarachnoid spaces and the venous sinus is also important to ensure cerebrospinal fluid drainage. This pressure gradient is between 3 and 5 mHg. Villus absorption of cerebrospinal fluid, either in the brain or in the spine, is a dynamic process which adapts the filtration rate of cerebrospinal fluid pressure. Arachnoid villi in the lumbosacral nerve roots increase cerebrospinal fluid absorption in the upright position in response to gravity, and the absorbed cerebrospinal fluid then enters the lymphatic system. Cerebrospinal fluid can also be absorbed by the olfactory mucosa and the cranial and spinal nerve sheaths, especially from the trigeminal, optical fascial, and vestibulo-cochlear nerves, the ependyma, and extracellular fluid according to pressure gradients. From the cranial nerve sheaths, it drains further in the lymphatic system. Absorption towards the interstitial compartments occurs via Virchow-Robin perivascular spaces. Cerebrospinal fluid stasis is associated with abnormal mechanical cord tension, vertebral subluxation, diminished respiratory function, increased sympathetic tone, facilitated spinal segments, tension in the dura mater. The reduction of cerebrospinal fluid turnover may contribute to the accumulation of metabolites seen in aging and neurodegenerative disease. Most important here is the free mobility of the trabecular structure in the subarachnoid space. More details on tests and techniques on this system can be found in the book on the cerebrospinal fluid and its influence on health. The tests and techniques there are described and showed in streaming videos. The whole strategy to treat the mechanical forces. Lung and heart system. Very important to start with that. The upper cervicals, the autonomic nervous system, the thoracic outlet, the subarachnoidal space, skull and spine area, and reduce chronic stress. Blood pressure normalization, drinking more than enough water, minerals, sleep. That's the whole strategy. There is a lot to do, and to have more details, you can read the book on the cerebrospinal fluid and its influences on health, because also there you will find all possible tests and techniques to treat that whole system. Or you can of course also follow one of my postgraduate courses. So, we are at the end of this webinar, and I really must thank you for your scientific attention. Thank you very much, and I will see you in one of my next webinars. Bye.